I'm going to turn now to tRNAs. Okay? So this is the structure of a tRNA shown as a clover leaf. It's not actually the true structure of a tRNA. And as I said, this is the adapter between the RNA information that's read and the next amino acid that will be added to a protein. There are 4 parts to it that are, well, actually 5 if you count this variable loop. The acceptor arm is the part of the tRNA to which, at its 3' end, the amino acid will be attached. The anticodon loop is critical because it makes base pairs with the codon, is actually interpreting the information. And then the Cy-U loop and the D loop are just 2 additional portions to the tRNA that are important for its recognition and maintaining its structure. And then, as I said, there's a little variable loop here as well. These are about 70 to 90 bases long, so they aren't substantially, they're very short transcripts. Again, you'll recall that they're made by RNA polymerase transcription, okay? And they include a lot of modified bases, okay? So one of the reasons these 2 loops are called what they're called is that they each contain canonical modified bases. So this is a pseudouridine. The Cy-U stands for pseudouridine, and this is dihydrouridine, and they're both modified enzymatically, and those modifications in many instances are critical for the function of the tRNAs. Okay, so what is a tRNA? What sort of components or similarities and differences can we see? So there's many tRNAs in a cell, and it turns out they have a bunch of things that are the same amongst all of them, okay? And when I talk about conserved properties here, I'm not talking about in a given organism. I'm talking about conserved in all organisms, bacterial, eukaryotic. So when I say this is evolved once, it's evolved once in a very specific way, and it's got to be extremely conserved in order to be maintained so that this central process doesn't mess up, right? So you can't make and it's not easy to make a mutation in these because it affects so many different proteins simultaneously. So if you have a bad proline tRNA, you are likely to mess up or inappropriately synthesize thousands of proteins simultaneously, right? So you can't really sort of accept that for a little while you evolve to accommodate that change. Okay, so what's conserved? So the first thing is this CCA, the last 3 nucleotides in every tRNA is CCA. Interestingly, there are a few of them where that gets added post-transcriptionally. I'm not going to go through this in detail, but there's a cool little enzyme that adds CCA in an untemplated way to tRNAs. Who knows why the cell decided to do that for those guys? Seems like it'd be easy enough just to have the right sequence in the tRNA in the first place. There's also conserved bases in several of these loops. So the Cy-U loop has a lot of conserved bases. Okay, 7 of them are found in all Cy-U loops, 4 bases in the D loop, and 1 in the anticodon loop. And obviously, the conserved base in the anticodon loop is not part of the anticodon. Okay, so it's either here or here. I don't remember exactly where it is. Okay, and then the number of base pairs in the arms are conserved in many instances. And in some ways, you can think of that as basically meaning that the length of these stems are the same in almost all tRNAs. So that's true in the D arm, the Cy-U arm, and the acceptor arm. Interestingly, the anticodon arm can be of different length. Okay, so there's a lot of things the same. There's also a lot of things that are different. Okay, so obviously, and most importantly, the sequence of the actual anticodon varies between each tRNA. Okay, this variable loop can come in sort of 2 size ranges. So-called class 1 tRNAs have about 3 to 5 bases, so the loop is relatively small in those. And class 2 tRNAs have much longer variable loops of 13 to 21 bases. There's something called the discriminator base. That's the base just before the CCA acceptor bases, and its identity is obviously not different in all tRNAs. It can only be 4 different things. The reason it's called the discriminator, however, is that in many instances, changing that base can have dramatic effects on the ability to charge or couple an amino acid to that tRNA. Okay, and in some cases, can even change the amino acid that's coupled. Okay, and then finally, there's lots of other sequence differences. So the question is, what are the conserved regions for, and what are the distinct regions for? So we've sort of already talked about why you'd want to have conserved things, right? So all of these tRNAs are involved in this fundamental process of adding amino acids to an ongoing protein synthesis reaction. And so that interaction needs to be conserved, and these sorts of conservation regions are responsible for that. Okay? So these basically allow the tRNAs to all be used equally well in the process of translation. They have to bind to the ribosome in the same way. They have to interact with accessory factors that deliver the tRNAs to the ribosome in the same way. And so you need this similarity. Now, of course, on the other hand, we want to put different amino acids on these different tRNAs, right? If we put the same amino acid, or if we randomly put amino acids on the tRNAs, and I'll illustrate this much more directly in a moment, we're going to get a mess. Okay? So all of these features are used to distinguish the different tRNAs from one another so that the right amino acid gets attached. And this is the responsibility of an enzyme called an aminoacyl tRNA synthetase. And I'm going to call this a AA tRNA synthetase for short. Okay? And these enzymes play a critical role in the fidelity of translation. Okay? So this sort of cloverleaf structure is a very easy way to look at a tRNA, but it actually only bears passing resemblance to what a tRNA really looks like. And you can envision the way the tRNA looks by sort of folding the tRNA. Okay? And in some ways, you can imagine that what's happened is that you've stacked the base pairs here on top of the base pairs in the anticodon loop. So this is the D loop. Okay? So now you have a continuous region of base pairing along here. And then you've done a similar thing with the Psi-U loop and the acceptor stem. There's no loop here, obviously, because it's not attached. And you get another region of continuous, in many ways, this looks a little like double-stranded DNA, okay, in each of these cases. So you get a helical turn in the same way as you do the other. And what this gives you is this L-shaped molecule, and you're able to now have a more compact form than this open sort of cloverleaf structure, which is really only used when you want to write out the bases and look at it as really discrete entities. This is actually a graphical representation of an actual tRNA structure. So the crystal structure of many tRNAs has been solved at this point, and this is what they look like. And you can see here's sort of a helical region here and a helical region here. And the main thing I want to point out here is how the anticodon loop, okay, including the anticodon here, is quite far away, is in fact on the opposite end of the molecule from the acceptor end where the amino acids are going to be coupled. And that's going to become very important because this part reaches into one part of the ribosome that decodes the message, and this part reaches into an entirely separate part. This is in the small subunit. This part that it reaches to is in the large subunit where peptide synthesis occurs. Okay? And so they, in much the same way as they link nucleic acid information to amino acid information, they also link the so-called decoding center of the ribosome, where you take that mRNA information and pass that on to the peptide synthesis center, or peptidyl transferase center is what it's actually called, in the ribosome at the other end. Okay, so they're reaching between these two states using the separation between the anticodon and the acceptor end.